In this video, we are going to look at the major topic, lipids. Lipids are a heterogeneous group of structurally related or unrelated and functionally diverse organic compounds. They are widely distributed in plant and animal tissues. In plants, they are present in large quantities in nuts, seeds, and fruits. In animals, they are stored in the adipose tissue, nerve tissue, and bone marrow. In general, lipids are nonpolar, hydrophobic, fatty, oily, or waxy compounds, insoluble or sparingly soluble in water, but readily insoluble in nonpolar organic solvents such as alcohol, acetone, ether, benzene, chloroform, etc. However, some short chain fatty acids such as butyric acid and caproic acid and mono and diglycerides having free polar hydroxyl groups are water soluble. So, molecular components of lipids. Lipids are formed of carbon, nitrogen, and oxygen, and occasionally nitrogen and phosphorus also. Cellular lipids are primarily esters of fatty acids and alcohols. Cellular lipids are primarily esters of fatty acids and alcohols. Esters of fatty acids and alcohols. Fatty acids in general are long-chain aliphatic organic acids. The commonest alcohol in both plant and animal cells is glycerol, but in some plant lipids it is replaced by cholesterol. So fatty acids are aliphatic hydrocarbon chains with a carboxyl group at one end. In other words, they are long, straight-chain monocarboxylic acids with the general formula CH3, CH2 raised to N, and COH. The fatty acid molecules consist of a long, nonpolar, acyclic or aliphatic and unbranched hydrocarbon chain linked with a polar and ionizable carboxyl COOH group at one end. Thus, fatty acids are amphipathic molecules since they contain both polar and nonpolar groups. The carboxyl group forms the head of the molecule and the hydrocarbon chain forms its tail. The hydrocarbon fatty acid chain has a backbone of carbon atoms to which hydrogen atoms remain attached. Most fatty acids are synthesized from two carbon C2 units acetyl radicals. So naturally occurring fatty acids have even number of carbon atoms ranging between 12 to 26, most commonly 16 or 18. Fatty acids with odd number of carbon atoms are occasionally found in small amounts in animal tissues, microbial cell wall, etc. In such cases, the number of carbon atoms ranges from 3 to 25. Examples are propionic acid, valeric acid, etc. Saturated and unsaturated fatty acids. There are two types of fatty acids, saturated and unsaturated. In saturated fatty acids, the maximum possible number of hydrogen groups remain attached to the carbon backbone. In them, adjacent carbon atoms are linked through single bonds, but in unsaturated fatty acids, some carbon atoms will not have the full complement of attached hydrogen atoms. So such carbon atoms share a double bond. Thus, fatty acids are saturated when their hydrocarbon chain contains only single bonds, and the unsaturated are those lipids when one or more of double bonds are present. Unsaturated fatty acids with only one double bond are called monosaturated fatty acids, and those with two or more double bonds are called polysaturated fatty acids. Unsaturated fatty acids melt at much lower temperature than do saturated fatty acids. Acetic acid, palmitic acid, stearic acid, butyric acid, lauric acid, myristic acid, etc. are saturated. Linoleic acid, linolenic acid, uric acid, oleic acid, and arachidonic acid are unsaturated. So they melt at much lower temperature. Okay. Triglycerides containing only saturated fatty acids are solids at room temperature, and those which contain only unsaturated fatty acids are liquids. Most plant fats are unsaturated and hence liquids at room temperature, so they are often called oils, but animals' fats tend to be saturated and solid. So that is something which is very particular about saturated and unsaturated fatty acids. Okay. So next topic is about essential and non-essential fatty acids. Based on nutritional essentiality, Based on nutritional essentiality, fatty acids are classified into essential or indispensable and non-essential or dispensable fatty acids. Essential fatty acids are the fatty acids which cannot be synthesized in animals and hence they must essentially be obtained from diet. On the other hand, non-essential fatty acids can be synthesized in animals and so they need not necessarily be obtained from diet. For human beings, linoleic acid, linolenic acid, arachidonic acid, icosapentaenoic acid and docosahexaenoic acid etc. are essential fatty acids, and stearic acid, palmitic acid, oleic acid, butyric acid etc. are non-essential fatty acids. EEA, that are the essential fatty acids, are long chain polyunsaturated fatty acids. PUFA, polyunsaturated fatty acids. Even though they are indispensable, they are required only in very small amounts, just as vitamins. They are important in membrane structure, cell growth, reproductive functions, and prolonged blood clotting. Also, they lower blood cholesterol level and thereby control hypercholesteremia, atherosclerosis, thickening of arterial walls, and coronary artery disease. So, deficiency of essential fatty acids in rats and some other animals have been shown to cause skin lesions, retarded growth, and problems related to pregnancy and lactation. Deficiency in human infants and children may cause skin lesions, eczema, phrenoderma, and toad skin, horny skin, eruptions on limbs, back, and buttocks, loss of hair, and poor wound healing. Fatty acids consist of both water-soluble polar and water-insoluble non-polar portions, so they do and do not dissolve in water. The water insolubility of fatty acids depends upon the presence of carboxyl head and the length of the hydrocarbon tail. The head is hydrophobic and hence responsible for water insolubility. Increasing length of the tail makes the fatty acid increasingly water insoluble. Usually fatty acids with more than 7 carbon atoms are water insoluble. Hydrolysis of triglycerides with NaOH or KOH is called saponification, soap formation. It yields a mixture of soap and glycerol. Triglycerides containing highly unsaturated fatty acids tend to undergo auto-oxidation in exposure to air. In this process, molecular oxygen attacks fatty acids having double bonds, yielding complex products which are responsible for the off-taste of rancid fats. Triglycerides containing highly unsaturated fatty acids tend to undergo auto-oxidation on exposure to air. In this process, molecular oxygen attacks fatty acids having double bonds, yielding complex products which are responsible for the off-taste of rancid fats. Glycerol will be there. So, glycerol is a straight chain trihydroxy trihydric alcohol CH2OH, CHOH, CH2OH, which can readily combine with weak acids. A glycerol molecule has 3 linkage sites for esterifying 3 fatty acid molecules. The linkage occurs between the acidic carboxyl group of one fatty acid molecule and the alcoholic hydroxyl group of the glycerol molecule with the elimination of one molecule of water. A hydrogen atom from the OH group breaks away and combines with the hydroxyl radical of the COOH group and forms one molecule of H2O. Consequently, adjacent carbon atoms link together through an oxygen atom. This type of linkage between an acidic group and an alcoholic group is known as an ester linkage. The products of the ester linkage between fatty acids and glycerol are known as glycerides, acyl glycerols, or glycerol esters. They are neutral fats. If a glycerol molecule is esterified by only one fatty acid molecule, the product is a monoglyceride, monoacyl glycerol, or monoester of glycerol. If it is esterified by two fatty acid molecules, the product is a diglyceride. If it has 3 fatty acid molecules, a triglyceride is formed. Triglycerols form the most abundant group of natural fats. So, glycerol, we will be drawing 3 carbons, 1, 2, 3. Then they are connected onto the left side by hydrogen and onto the right side to OH group. Then we will be putting a hydrogen on the top carbon and another hydrogen on the bottom carbon. So that is the structure of Glycerol. There are three OH groups which are situated on the right side. So they are linkage site for fatty acids. This is the structure of glycerol. These are the linkage sites for fatty acids. Okay. Three fatty acids, glycerol, triacyl glycerol will be there. So three fatty acid moles plus one glycerol mole is equal to 1 triacyl glycerol mole plus 3 H2O moles. Ester linkage between fatty acids and glycerol. Now we have already learned about the structure of glycerol. So what are the major points about glycerol? Glycerol is a straight chain trihydroxy alcohol. Glycerol is an alcohol, nothing else. It's an alcohol which can readily combine with weak acids. Glycerol readily combine with weak acids. The glycerol molecule has three linkage sites for esterifying three fatty acid molecules. There will be forming an ester bond. What is an ester bond? The linkage occurs between acidic carboxyl group of the fatty acid molecule and the alcoholic hydroxyl group of the glycerol molecule. COOH of fatty acid molecule and OH of the glycerol, with the elimination of one molecule of water. That is something which is termed as a ester linkage. Now we are going to learn about the classification of lipids. There are three major groups of lipids, namely simple lipids, compound lipids or conjugated lipids or heterolipids, and derived lipids. So first one is the simple lipids. Simple lipids or homolipids are alcohol esters of fatty acids which yield fatty acids and an alcohol on hydrolysis that most probably will be a glycerol. They are formed of carbon, hydrogen and oxygen. In fact, they are neutral lipids formed of lipid monomers linked to alcohols through ester bonds or amide bonds. Amide linkages. They include neutral fats and waxes. Fats are esters of fatty acids and glycerol, so they are also called acyl glycerols or glycerides. On the other hand, waxes are esters of fatty acids and non-glycerol aliphatic alcohols such as myrcyl alcohol. Fats are fatty acids and glycerol, but waxes are fatty acids with non-glycerol aliphatic alcohols. Okay, so now first learn about the neutral fats. Fats are mixtures of triacyl glycerols, formerly termed triglycerides. A triacyl glycerol in turn is an ester of fatty acids and glycerol. It is a fully esterified glycerol formed of three fatty acid molecules linked on one glycerol molecule. Triacyl glycerols containing the same kind of fatty acid in all the three positions are called simple triacyl glycerols or homoglycerides. Examples are triesterin, tripalmitin and triolane. This contains stearic acid, palmitic acid and oleic acid respectively. Triacyl glycerols containing 2 to 3 kinds of fatty acids are called mixed triacyl glycerols or heteroglycerides. Most natural fats are mixtures of simple and mixed triacyl glycerols. In triacyl glycerols, free acidic and basic groups are absent. So fats are often called neutral fats. Neutral fats, glycerides, acyl glycerols or glycerol esters are the products of the ester linkage between fatty acid and glycerol. Triacyl glycerol form the most abundant group of neutral fats. Next is the waxes. Waxes are mostly esters of long-chain fatty acids and long-chain non-glycerol aliphatic and monohydroxy alcohols. They may contain dihydroxy alcohols, free fatty acids and saturated hydrocarbons also. In brief, they are mixtures of esters and ethers derived from higher fatty acids and higher monobasic alcohols. Waxes are highly hydrophobic and resistant to atmospheric oxidation. They form a water repellent protective coating, grease on the leaves and fruits of plants, skin and fur of animals, feathers of birds, and the exoskeleton of insects. Also, they form a constituent of beeswax. Waxes protects plants from water loss and abrasive damage. Some animal oils, such as sperm whale oil, are formed mostly of waxes rather than by fats. Beeswax, carnauba wax, sperm whale wax, linolein, lanolin, etc. are some common forms of waxes. Now let's learn about beeswax. This is a mixture of esters, free fatty acids, alcohols, and hydrocarbons. Its major constituent is merusyl palmitate, which is an ester of merusyl alcohol and palmitic acid. Next is the carnauba wax. This is a plant wax found in leaf cuticle. It is also a complex mixture of esters, free fatty acids, alcohols, and hydrocarbons. Next is the sperm whale oil. This is the oil from the head of sperm whale. Nearly 75% of it is a wax ester and 25% triglycerides. Saturated and unsaturated fatty acids and alcohols are also present. Then lanolin or wool fat will be there. This is the protective coating over wool fibers. It is a wax rather than a fat. It is complex mixture of free and esterified cholesterol and esterols, lanosterol and agnosterol. Lanolin can take up water without dissolving. This property makes it valuable as a medium in the preparation of ointments and cosmetics. That's the peculiarity of wool fat or lanolin. That's a kind of wax which are seen as a protective coating over the wool fibers. So this property, they have a particular property which is very peculiar and that can be used for preparation of ointments and cosmetics. The property is that they can absorb water without dissolving into that water. So that is a particular property of this lanolin. So we just classify lipids into major three parts like simple lipids, compound lipids, and derived lipids. Again, we will be classifying the simple lipid into neutral fats, oils, and waxes. Then the derived lipids into prostaglandins and isoprenoids. The middle one, that is the compound lipid, we have transferred that into four different categories like phospholipids, sulfolipids, glycolipids, and lipoproteins. And again, the phospholipid we are classifying into three like phosphoglycerides, sphingolipids, plasmalogens, cardiolipin. And then the glycolipids are cerebrosides and gangliosides. Now that's the overview of classification. Now let's learn about the conjugated lipids, compound lipids or heterolipids. Compound lipids or heterolipids are the lipids which remain linked with non-lipid molecules such as protein, oligosaccharide, and phosphates. So on hydrolysis they yield fatty acid, of course fatty acid, alcohol, that's quite natural, and other compounds. Lipids contain atoms other than carbon, hydrogen, and oxygen, most notably phosphorus, sulfur, and nitrogen. Phospholipids, glycolipids, sulfolipids, and lipoproteins are examples. Phospholipids are significant in the organization of cell membrane. Glycolipids and lipoproteins are also associated with biomembranes. So first one is the phospholipid. Phospholipid, from the name we can easily say that this is a combination of heterogeneous group of phosphate and lipid. Phospholipids or phosphatides are heterogeneous group of polar lipids. They are widespread among bacteria, plants, and animals. Most of them are derivatives of phosphatidic acid. Phospholipids are so named because they contain phosphoric acid or phosphate group. Other key constituents include glycerol, fatty acid, nitrogenous base, and alcohol. Phospholipids are amphipathic molecules with a polar and hydrophilic head and a nonpolar and hydrophobic tail. The head is formed of a negative phosphate group and a positive alcohol group, and the tail is formed of fatty acids. Glycerol forms the core constituent of phospholipids. There are two major groups of phospholipids, namely phosphoglycerides and sphingomyelins. Apart from this, there are some minor phospholipids, also example quoted as plasmalogens and cardiolipin, etc. So that is something which is very relevant about the phospholipids. Okay. Next is the Phosphoglycerides. Phosphoglycerides or glycerophosphates are the commonest phospholipids. This is a type of phospholipid, which are the phosphoglycerides. Phosphoglycerides. They are fatty acid esters of glycerol phosphate. A phosphoglyceride molecule is a diglyceride formed of two fatty acid molecules, a glycerol molecule and a phosphorylated alcohol complex called phosphate group complex. The phosphate group complex in turn consists of a phosphate group and an attached alcohol. The phosphoglyceride without an attached alcohol is called phosphatidic acid. Polar head will be there and glycerol will be there, and non-polar tail will be there. Fatty acid we can see, phosphate will be there, and phosphate group complex can be seen. So that is something which is very important about the structure of phosphoglyceride molecule. Based on the attached alcohol, several kinds of phosphoglycerides can be recognized. Examples are phosphatidylcholine, choline phosphatide, phosphatidyl ethanolamine, ethanolamine phosphatide, phosphatidylserine, serine phosphatide, phosphatidyl inositol phosphatide, etc. This contain the attached alcohols choline, ethanolamine, serine and inositol respectively. Of this, phosphatidylcholine, lecithin and phosphatidyl ethanolamine are very significant in the organization of protoplasm. So the phosphoglyceride molecule, we will be drawing a polar head and a glycerol, and non-polar tail will be there. Two fatty acids are joined. Next is the lecithins, phosphatidylcholines. Lecithins are choline derivatives of phosphatidic acid. So on hydrolysis, they yield glycerol, fatty acids, phosphoric acid and choline. They contain the acidic COOH group of phosphoric acid and the basic OH group of choline. So they exist as salts or zwitterions. They are highly polar molecules capable of orienting themselves in a definite fashion. This capacity for orientation enables them to play a significant role in the structural organization of protoplasm and cell membranes. Phosphatidylcholine is the storage form of choline within the body. Next is the cephalins, phosphatidyl ethanolamines. Cephalins are ethanolamine derivatives of phosphatidic acid. They are structurally similar to lecithins except that choline is replaced by ethanolamine, cholamine. They also form internal salts or zwitterions and play a major role in the organization of protoplasm and cell membrane. Cephalins are particularly abundant in brain. Cephalins, we can easily write this point that they are particularly abundant in brain because cephalic region means head region. So cephalins are particularly abundant in brain. Lecithins are also a type of phosphatidylglyceride. Sphingolipids. Sphingolipids or phosphingosides are the derivatives of spingenin, formerly called sphingosine. They do not contain glycerol. Spingenin is a long-chain unsaturated nitrogen-containing amino alcohol. Sphingolipids contain phosphoric acid, fatty acids, spingenin, and a nitrogenous base. The commonest base is choline. There are two groups of sphingolipids, namely sphingomyelins and ceramides. Sphingomyelins are the common type of sphingolipids. They are found in the plasma membrane of most cells and are especially abundant in the myelin sheath of nerve fibers, hence the names sphingomyelins. Ceramides are the simplest type of sphingolipids. They consist of a fatty acid molecule linked to the amino group of sphingogen by a amide linkage. Ceramide acts as a second messenger signaling molecule in regulating apoptosis. Ceramide acts as a messenger signaling molecule in regulating apoptosis. So, programmed cell death, cell cycle and cell differentiation. Sphingenin and ceramide is also there. In animals, sphingolipids are abundant in blood, plasma, erythrocyte envelope, brain and nerve tissue. In small amounts, they are found in other tissues, lung, liver, kidney, spleen also. They form an essential constituent of the myelin sheath of the medullated nerve fibers. Just as lecithins and cephalins, they are also constitute the essential part of protoplasmic structure. Sphingolipids are abundant in blood plasma, erythrocyte envelope, brain and nerve tissue. In small amounts, they are found in other tissues, lung, liver, kidney, spleen, etc. also. They form an essential constituent of the myelin sheath of medullated nerve fibers, just as lecithins and cephalins. They also constitute the essential part of protoplasmic structure.